Hi, I'm Stan Sugrue, and I do customer training and also some internal training here at the Beckman Coulter Technology Center in Miami. And we're going to talk about today is the Multisizer, which we normally refer to as the MS, so this is the BS before the MS. We're going to let you guys know and gals know what is it that people use this instrument for. But first we want to talk about how does this thing actually make a measurement. The Coulter Principle is something that was developed by Wallace Coulter and patented by him in the early 50s, but really the invention was resolved in the late 40s. And it was a very eloquent solution to the individuals who had to slave over microscopes staring into small optical fields and count cells for hours at a time. And it was a very rapid, labor-saving device, and that's where it was originally marketed to. But the principle itself is applicable to any particle within a size range of 0.2 microns all the way up to 2,000 microns currently. And the size range itself is determined by the sensor in the system, which we refer to as aperture tubes, and the way the technology actually makes a measurement is very, again, eloquent in its simplicity. We've got an aperture tube which has an internal electrode, and it's placed into a conductive medium, something that has some level of ionic strength so it can carry a current. And there is an external electrode in that medium. Now the tube itself has a hole at the bottom, that's the quote aperture, of a very well-defined size, and the aperture is normally referred to by the size of the aperture. So a 100 micron aperture has a 100 micron hole in the bottom of the aperture, through which when a vacuum is applied to the tube itself and then pulled into a distribution and waste system, it pulls the sample of whatever particles you have that are suspended in the sampling vessel through this hole in the tube. And as each particle passes through the hole in the tube, the instrument is measuring the change in electrical current that's flowing between that internal electrode inside the tube and the external electrode, which is in your sample. This principle is very simple, that as a particle that is non-conductive passes through this zone, which is being measured as far as current, it acts as an insulator, and therefore the current drops. And this pulse that's measured as a voltage pulse is telling us that a particle has passed through that zone. Furthermore, what makes it so useful as a method is that this pulse displacement of current is directly proportional to the volume of the particle that passed through the sensing zone. So what it does in the final dance between the particle and the instrument is that it has passed through this sensing zone and been detected and counted and sized simultaneously. So the instrument actually counts every particle that passes through the aperture. So it's a quantitative, high-resolution particle sizing instrument. And all it requires is that your particles that you're trying to measure are compatible with the size of the aperture tube. The aperture tube has a physical definition of what it can handle and measure accurately, and that's 2 to 60% baseline of the aperture tube itself is the size range. So if you have a 100 micron aperture, you can measure from 2 to 60 microns with that aperture. If you had a 1,000 micron aperture, you would then be able to look at 20 to 600 micron samples. So 2 to 60%. It also has an extended range which allows you to additionally recover another 20% of the particle material that goes through. But we're not going to discuss that, but there is an extended range so that you don't, having maybe heard about this, you say, well, why did he say 2 to 60? It actually will give you data on 2 to 80%, but in a single measurement, it will give you 2 to 60%. So this means that all you do is adjust your aperture selection to whatever the size range of the sample that you're required to look at. And this will allow you to get high resolution particle sizing as a function of volume, as well as a highly accurate count or concentration of what the sample population is.